the most abundant biomolecules on Earth. They are found everywhere. Carbohydrates are a major source of energy for all living organisms, such as animals and plants. But they are not only important for energy, carbohydrates also serve as important structural components. For example, DNA contains the carbohydrate ribose, and the plant cell wall are made up of the carbohydrate cellulose. Carbohydrates mainly contain carbon, hydrogen, and oxygen atoms in a molar ratio of 1 to 1. 1 carbon, 2 hydrogens, and 1 oxygen. Carbohydrates can be divided into 4 types. These are monosaccharides, disaccharides, oligosaccharides, and polysaccharides. The word saccharide is derived from the Greek word for sugar. Now let us look at each of these types of carbohydrates and learn a bit more about their structure and how they are formed. This is a biochemistry lesson. Let's begin with monosaccharides. Monosaccharides are also referred to as simple sugars, and they are the smallest units that make up any carbohydrate. They are the building blocks. The 3 main monosaccharides in the human diet include glucose, galactose, and fructose. Now these structures may look intimidating, but all you need to know is that they contain carbons, hydrogens, and oxygens. Let's look at glucose first, which you all probably have heard of. Glucose is the main source of energy for humans. Here you can see glucose in its cyclic chemical form. What's important to know about glucose is that it contains 6 carbon atoms. So let's have a closer look. Here, C represents a carbon atom. And we can label these carbon atoms of glucose with numbers 1, 2, 3, 4, 5, 6 in this specific order. So glucose has 6 carbon atoms. This particular type of glucose is actually an alpha glucose because it has an alpha configuration. Alpha carbohydrates is where the hydroxyl group, the OH group of carbon number 1, is pointing in the opposite direction to the carbon number 6. So these are opposite each other. And there is another type of glucose known as a beta glucose. This is essentially where the hydroxyl group of carbon number 1 and carbon number 6 are pointing in the same direction. So beta carbohydrates is where the hydroxyl group of carbon number 1 is pointing in the same direction as carbon number 6. And again, these alpha and beta carbohydrates, they also apply to other types of carbohydrates, such as galactose as well as fructose. So for example, this galactose molecule is actually a beta galactose because the hydroxyl group here and the carbon number 6 is pointing in the same direction. Similarly, this fructose here is actually in a beta configuration, so it's a beta fructose because the hydroxyl group here is pointing in the same direction as carbon number 6. So I hope you understood the structure of the 3 major monosaccharides in the human diet. Now let's look at disaccharides. Disaccharides are made up of 2 monosaccharides. So for example, a glucose molecule and another glucose molecule can form a bond with each other. This new disaccharide is called maltose. Maltose is essentially 2 glucose molecules linked together. It is linked together by an alpha 1,2,4 glycosidic bond. It's called alpha 1,2,4 glycosidic bond because carbon number 1 of this glucose and carbon number 4 of this glucose are involved in the linking process. And it's called alpha because both these glucose molecules are in an alpha configuration. Now the process of linking monosaccharides with one another is called condensation, and here water is released. Therefore, the reverse reaction is hydrolysis, and this is where we add water. Adding water to an alpha 1,2,4 glycosidic bond will break the bond. So maltose is only one example of a disaccharide. Let's look at some other common examples. Now this galactose molecule can link with a glucose molecule. So this particular glucose molecule is actually in a beta configuration because remember, the hydroxyl group here is pointing the same direction as carbon number 6. So this galactose molecule and the glucose molecule can form a link, and through the condensation process it will form lactose. Lactose is made up of galactose and glucose. The galactose and glucose is linked together by a beta 1,2,4 glycosidic bond. Now it is called a beta 1,2,4 glycosidic bond because the galactose and glucose are in a beta configuration, and also carbon number 1 and carbon number 4 are involved. The reverse reaction to break lactose requires hydrolysis by adding water. Now lactose, as you all probably know, is found naturally in milk. The third type of disaccharide I want to talk about is where we form a bond between one glucose molecule and one fructose. This glucose molecule is an alpha glucose because as you can see, the hydroxyl and carbon number 6 are pointing in the opposite direction. So glucose and fructose can form a link, and through the condensation reaction, removal of water, it can form a disaccharide called sucrose. Sucrose is made up of one glucose and one fructose. The bond between the glucose and fructose is a little more complicated, as it is a glucose alpha 1 and fructose beta 2 bond. This sort of linking occurs. So the glucose is in an alpha configuration and the fructose is in a beta configuration. And it's carbon number 1 of glucose and carbon number 2 of fructose that are involved in the linking process. So essentially, fructose flips over. Sucrose, as you all know, is table sugar and is formed by plants and not formed by animals. So humans cannot form sucrose. Sucrose is broken down through a hydrolysis reaction. So the disaccharides, maltose, lactose, and sucrose are all good examples that we encounter in our normal diet. Now let's look at oligosaccharides. Oligosaccharides basically consist of short chains of monosaccharides, typically less than 20 monosaccharides linked together. Actually, a disaccharide can be referred to as an oligosaccharide. Now let's look at an example of an oligosaccharide. So if we were to take this maltose and add another glucose molecule to it, through a condensation reaction again, we can form an additional alpha-1,2,4 glycosidic bond. This oligosaccharide is called maltotriose, tri as in 3. And this maltotriose is made up of glucose, and they're linked together, as I mentioned, by alpha-1,2,4 glycosidic bonds. To break down these bonds requires hydrolysis, the addition of water. And this structure can keep growing with the addition of more glucose molecules. But when the oligosaccharide eventually exceeds 20 monosaccharides with 20 bonds, the carbohydrate is then referred to as a polysaccharide. So from an oligosaccharide, it becomes a polysaccharide. Most carbohydrates found in nature occur as polysaccharides. Polysaccharides are also known as glycans. To simplify things, polysaccharides can be a homopolysaccharide or they can be a heteropolysaccharide. A homopolysaccharide means the polysaccharide only contains a single type of monosaccharide. For example, it only contains glucose molecules linked together. A heteropolysaccharide means that the polysaccharide contains 2 or more different monosaccharides. So for example, a long chain of fructose and glucose molecules. To make things a little bit more interesting, a polysaccharide can also be unbranched, like what you see here, or it can be branched. This goes for both homopolysaccharides as well as heteropolysaccharides. Heteropolysaccharides can also be unbranched or branched. What you have to understand is that polysaccharides I am currently drawing are very small. In reality, the polysaccharide contains thousands, are made up of thousands of monosaccharides linked together. In this section of the video, we won't focus on heteropolysaccharides, but we will look at homopolysaccharides. Because homopolysaccharides serve as storage forms of monosaccharides in both humans and plants, and even bacteria. So they're very, very important. So let's look at some examples of homopolysaccharides. Starch is a storage form of monosaccharides in plants. Starch is the main carbohydrate in the human diet and are found in our bread, cereal, and rice. Starch is only made up of glucose because it is a homopolysaccharide. Let us zoom into this area here and learn a bit about the bonds. So here we have our regular alpha-1,2,4 glycosidic bond between 2 glucose molecules. And this is because carbon number 1 and carbon number 4 of these glucose molecules are involved in the linking process. However, the branching point here is actually an alpha-1,2,6 glycosidic bond between 2 glucose molecules. And that is because carbon number 1 of this glucose and carbon number 6 here are involved in the linking process. So what to take out of this is that starch has 2 forms, it can be branched, like what I just explained, or starch can be unbranched. If starch is unbranched, so it is only a chain of glucose linked together by alpha 1 to 4 glycosidic bonds, it is referred to as amylose. If starch is branched, it contains both alpha 1 to 4 and alpha 1 to 6 glycosidic bonds between glucose, and therefore it is referred to as amylopectin. Amylose and amylopectin are two forms of glucose polymers. The other good example of a polysaccharide is glycogen. Now, glycogen is a homopolysaccharide because it is made up of glucose. Glycogen can also be branched or unbranched. Glycogen is a storage form of glucose in animals, such as humans. Starch and glycogen are actually very similar in structure. They both are made up of glucose, and they can either be branched or unbranched. So both starch and glycogen contain amylose and amylopectin. The only difference is that glycogen has these branch points occurring every 8 to 12 glucose residues. In starch, these branch points occur every 24 to 30 glucose residues. So the branch points occur more frequently in glycogen than starch. And this, of course, will influence the structure in some way. Another type of polysaccharide is dextrans. Dextrans are structural components in bacteria and yeast. These polysaccharides are made up of alpha 1 to 3 and alpha 1 to 6 glycosidic bonds. So here we have glucose units with alpha 1 to 3 bonds and alpha 1 to 6 bonds. However, the dextrans can also contain alpha 1 to 2 and alpha 1 to 4 glycosidic bonds. Finally, the other polysaccharide worth mentioning is cellulose. Cellulose are structural components in plants. They make up the plant cell wall. They are unbranched homopolysaccharide consisting of thousands of glucose molecules. So here you can see unbranched cellulose on top of each other. In cellulose, the glucose molecules have a beta configuration. And therefore, the bonds between these glucose molecules are beta bonds. The glucose molecules are linked together by beta 1 to 4 glycosidic bonds. Humans do not have enzymes that break down, that hydrolyze beta 1 to 4 glycosidic bonds of cellulose, and so humans cannot digest cellulose. Now, even though cellulose are only chains of beta glucose, so unbranched beta glucose, these chains can form hydrogen bonds with each other, forming a very strong structure.